My apologies to the participants of this symposium. I wanted very much to come and hear what you have to say. Unfortunately, I had to cancel at the last minute. What I want to do here is tell you about a new method for seeing the dynamics of a cognitive process. I believe this is a new approach that holds great promise for the future. Usually we are used to see images like this with fMRI, with one or two locations which are active during a given process. Nowadays, it is common to speak about network of processes, such as the default networks or the tinnitus networks. However, you have to remember that a simple mental process, like comparing shape with color, consumes only 30 milliseconds, while even a simple cognitive process consumes 500 milliseconds. So there are many elementary mental processes in a complex cognitive process. And we wish to see the development in time. So our working hypothesis is that any cognitive process involves many cortical areas. Activations of some may co-occur or develop in some specific order. And the aim of this talk would be to show that such sequences may be detected and to illustrate such specific sequence which is specific to one cognitive process versus the other. Imagine that for a given process we can see activity starting here and then added here and then here, here and here and so on. The movie shows such a sequence of cortical activation which are specific to routine tapping with the fingers while listening to music. In this movie, we see the subject brain as rendered from fMRI. It is partially transparent, so you can see also brainstem and inner structures. And you see this point started, now this point, now the three red points are added, and so on. You note that at the beginning, all the points are only on the left hemisphere. And it takes a while before some points appear also on the right hemisphere, like here and here and here. This movie is slowed down 1,000 times. So down here you see the actual real time in milliseconds. You see that the resolution of the events here is much faster than you can hope to see with fMRI. When several points are activated together, they appear in the same color. So one can appreciate the order and type and number of points which are recruited into the process. These things go on and on. The entire length here would be something like 90 milliseconds for this particular process. If one could detect such sequences of cortical activation that are related to any cognitive process, it would have huge impact on how we study and understand brain mechanisms of cognition. In this talk, I want to illustrate how such specific sequences of cortical regions were identified in a test case. To do this, we need to understand when a region in the cortex becomes involved in the process. This detection is based on two claims. Claim 1. Sharp transitions in brain slow wave activity like EEG, electrocardiogram, etc. are associated with sudden increase in neuronal population activity. We use data from microelectrode recording in cortex of behaving monkeys to extract two signals. By low-pass filtering, we obtain the local field potential, LFP. By high-pass filtering, we obtain spiking from multiple neurons, multi-unit activity. In the LFP, we look for brief transients such as this one here. We call them mini-evoked responses because they look like a regular evoked response but smaller. We have shown that there is a close association between such mini-evoked responses and brief increases in the multi-unit activity rate. so they might signify increase in activity of neurons. Claim 2. Similar mini-evoked responses may be found also in MEG recorder in human subjects. So this is the situation. We record brain activity with magnetoencephalogram with 248 magnetometers. This is the positions. The black stars are the positions of the magnetometers. The blue dots are dots on the skull of the subject. Here is the nose. And the red dots are dots on the surface of the cortex as rendered from the MRI of this subject. Stephen Robinson from Neuro-Imaging developed the software to reconstruct the activity in several points in the cortex based on the MEG. We can therefore look on this cube of points. The center point is the point of interest, and around it are 8 points at the vertices of the cube at distances of 2 by 2 by 2 centimeters. We reconstruct the activity in all 8 points and the center simultaneously for 27 orientations of the cube. We then select the orientation which has smallest correlation between the center and the vertices, and we call this the cortical current dipole amplitude at this point. Here is an example how things would look. The blue trace is the amplitude of the transient in the cortical current dipole amplitude. The red trace is similar from LFP of intracortical recording with microelectrodes. You see the great similarity of the sharp transients in two. You have to note that the amplitude of the blue trace here is given in standard deviations. So this is quite small, and there is a real chance of missing such a transient in noise. So we do have some fraction, unknown fraction of false negatives. Also, one may expect that in noise there would be also things of that sort. Indeed, if we take noise, filter it like the MEG, and then look for transient like this, we do find transient like this also in noise. But with the detection threshold that we used for MEG, the rate of this spurious, noisy transients is 1/3 of that in the real data. So we can estimate that 1/3 of what we detect are false positives. Here is the experiment. The subject listens to drum beat, something like boom, tick, boom, tick, boom, tick, or boom, tick, tick, boom, tick, tick. He is asked to tap with 2 fingers to the rhythm of the drum beats. So he taps like second, third, second finger, third, or second, third, third, second, third, third. From time to time, the rhythm is changed from 2/4 to 3/4. When this happens, the subject typically misses 1 or 2 beats and then resynchronizes with the new beats. Within this behavior, we are interested in 2 types of events. The last congruent beat, which can be like this, or the first incongruent beat that can be like this or like this. Note that both the same and change include hearing primary and secondary drum beats and second and third finger taps, but the mental process following the stimulus are quite different. From the MEG, we reconstructed the current dipole amplitudes over 550 points on the dorsolateral cortex. This cortical current dipole's amplitude, in this cortical current amplitude, we detected mini-evoked responses at about 3 per second per channel. the times of these mini-evoked responses were considered a point process, so we obtained about 550 parallel point processes. In them, we looked for precisely repeating sequences such as A, after T1 followed by B, after T2 followed by C, etc. If structures like this, A, B, C, D, etc., with the same delays repeated themselves twice or more, we counted them as precisely repeating sequences. We found many thousands repeating sequences in data following congruent bits and different ones in data following incongruent bits. Are the repetitions truly precise? We use a method of Bienenstock and Giemann that states as following. If no precision was there, then we may jitter every event by plus minus W millisecond, and it would not affect the statistics of what we see. So we do this here now. What I show is jittering within plus minus one sample every event, and again looking for the number of patterns that we find. This is the situation. This is the distribution of jittering the event times and finding number of patterns again and again. So you see here a distribution. This is what was found in the real data. Clearly, this is highly significant. Maybe something like this can be found also in random data. So we repeat the thing with random data. We change the detection threshold until we get about equal number of repeating sequences in the random data. But when we jitter the timing, you see that the distribution is right around where the center was. So in random data, we do not see anything precise. In real data, we see high precision. We found many thousands of precisely repeating sequences in data following the congruent bits, and different ones following incongruent bits. However, each of them repeated only 2, sometimes 3 times. This is strange, because if the same brain process repeated again and again, we expected to see the same sequences repeating again and again. Why didn't we see? Remember that we can easily miss a mini-evoked response, and we may have 1/3 of the detected ones be due to noise. These situations will badly interfere with our current algorithm of detecting precisely repeating sequences. But it could be that there is some substructure that remains. So there is something big, but every time we see only a fraction of it mixed with other things. To test this, we looked for triplets that appear, triplets of events that appeared in many of the detected repeating sequences in one conditions and not in the other. Indeed, we could do this. In this case, we saw about 240 pieces, triplets, that appeared in sequences in the incongruent, following the incongruent bit, but not following the congruent bit. If we take just 2 pieces of data unrelated to behavior and repeat the whole process, we also can see some specific triplets. But if we do this again and again, this is the distribution of number of specific triplets we see. Again, what we found is far out of what you can expect just from data which is not related to behavior. Now we can use it to superpose patterns in which the same triplet occur. Assume that the triplet A, B, C once is seen in the structure X, A, B, Y, C, Z, and another time in the structure U, A, B, V, C. We superpose them to get X, U, A, B, Y, V, C, Z, and so on. We can repeat this again and again until no more superpositions of similar pieces can be done. In this way, we obtain between 13 and 30 super sequences that seem specific to congruent bits and others that seem specific to incongruent bits. Now, are these really specific? To test this, we build an analog structure as follows. We take the shape of the mini-evoked response of this channel after the delay, like in the sequence, and for this channel in delay of this channel in the sequence, and so on. I've put here only 4 of them, but usually there would be 10, 20, 30 or more of them. But this would be too complex to illustrate, so we look here only on 4. Now this is a template. We place this template over the data, and then we correlate the piece of data, like in here, with the shape of the mini-evoked response for this channel at this time. We add all the fits for all the channels by first converting them to Fisher-Z transform and then adding them. And down here you see the result of this addition. Now we slide this set of windows over the data and compute sort of continuously this fit to the templates, and we get something like this, undulating things. Sometimes there is a clear peak in the fit to the data, and we mark this as a point of fit for the templates to the data. If we now look on all the trials, we see this picture. Time 0 here is the time of switch from 2/3 to 3/3, or from 3/3 to 2/3. Red ticks are points of fit to the sequences that were specific to the incongruent bit. Black ticks are points of fit to the sequences specific to the congruent bit. We can see that there is large excess of red bits after 0 and large excess of black ticks before 0. Now, does this overfit the data? We actually use the data to find the sequences, and now we see the sequences there. But not all the time was used. For the incongruent bit, we used all the pieces of data in this pink rectangle. But we see many red ticks also outside this. Also note that the red ticks start mostly 100 milliseconds after the change of the meter. For the congruent bit, we used either data from this blue rectangle or from this green rectangle, but we find excess of fits also in other times. So it's clearly that we don't have really bad overfitting of the data. Now let's look at a sequence which was specific to the incongruent bit. So in this movie, we'll see sequences of cortical activation specific to the entraining to a new musical meter. Again, like before, we see the brain of the subject semi-transparent. When the system starts, we see these 4 points appear, then this point, then later on at other points. You can see that already from the beginning the activity is distributed among the 2 hemispheres. Again, note that the time is slowed down 1,000-fold, and what we see here, the time in the experiment in milliseconds. Again, things that happen simultaneously appear in the same color. Each patch here is about 2 centimeters in diameter because this is the accuracy of the MEG localization. We don't mean that this entire region become active. We mean that within such a region, enough neurons become active to be seen as a short transient in the MEG recording. The thing develops again and again, and it is clear that this is very complex. But who says that the cognitive process is something very simple? The complexity of things promises great room for analysis of what goes in the brain. So in A and B, we see all the patches that were in the sequence that were specific to the congruent bit and in B for the incongruent bit. Here you see many of them concentrated on the somatosensory strip, also in the cerebellum, but also in other places. For the incongruent bit, we see many in the prefrontal and also orbitofrontal, some in the posterior parietal area, and some in the temporal area. One may tempt to call these networks of places associated with congruent or incongruent bits. But recall that what we are seeing is not just the position of these things, but also their exact sequence of their occurrences. So in conclusion, we demonstrated that it is feasible to find sequences of cortical events which are specific to one set of cognitive process or to another. Thank you. I hope you could see that there is a great promise in doing something like this.